In this video, we are going to look at paclitaxel and its effect on mitosis in cancer cells. We will start by asking, what is paclitaxel? Paclitaxel is a common chemotherapy that is used to treat many different cancers. This includes breast, lung, and ovarian cancers. Paclitaxel is a spindle poison. This means that it attacks microtubules during mitosis. Here we show the chemical structure of paclitaxel. And with this, we move on to mitosis. Mitosis is a process that happens in every eukaryotic cell. During this process, one diploid cell divides into two identical diploid daughter cells with accurately separated chromosomes. Mitosis happens in distinct stages, starting with interphase, then prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis starts with the cell in interphase. DNA duplication happens at this stage in preparation for the rest of mitosis. Entering prophase, inside the nucleus, the nuclear DNA is condensed into sister chromatids, with each pair being held together by attached cohesin loops. The cohesin loops bind in many places up and down the chromosome, keeping sister chromatids in pairs. Once chromatids are paired up, the nuclear envelope dissipates. A closer look at the spindle fibers show they are made up of tubulin fibers arranged in a tube-like shape. They consist of alpha and beta tubulin. Prometaphase begins as the spindle fibers form, extending towards the chromosome. The spindle apparatus is made up of the centrosome and the finger-like spindle fibers. The spindle begins to grow towards the chromatid pairs. While this occurs, kinetochore attachments bind to the centromere, ready for microtubule binding. Eventually, the spindles grow to meet the kinetochore attachments on the centromere and bind to them. During metaphase, the chromosomes are aligned on the equator of the cell, ready for the transition to anaphase. In order for this transition to occur, cohesins need to be unbound, releasing the chromatid sisters from each other. This is done using a protein called separase. Separase severs the cohesin arms from each other, releasing the chromatids. In anaphase, the chromosomes are each pulled to their respective poles. This is achieved by two mechanisms, known as anaphase A and anaphase B. Anaphase A involves microtubules. The microtubule fiber begins to strategically break down at the kinetochore end. This pulls the chromatid at the centromere. Simultaneously, the centrosomes are pulled by microtubule structures towards the cell membrane closest to the pole. Once the chromosomes have reached the pole, the spindle breaks down. Around the DNA structures, the nuclear membrane reforms. The cells then split down the membrane into two identical daughter cells, in a process called cytokinesis. So how does this relate to cancer and paclitaxel? Cancer cells divide rapidly and have different cellular properties. Paclitaxel targets these rapidly dividing cells and disrupts mitosis. It does this by interacting with microtubules. Paclitaxel binds to a binding site on beta tubulin, stabilizing the microtubules so they cannot break down during anaphase A. This leaves the cell stuck in metaphase, leading to apoptosis, a controlled cell death. This eliminates the cancer cell.